Hi, today we're going to talk about viruses. So we're going to talk about their structure, their function, we'll do a phenomena study about a virus, and then we'll have a CER prompt that you'll work on independently. So this is not going to be a comprehensive viral review for obvious reasons. This is just our introduction stage of things. So yes, there will be more details that we'll cover later, but for the most part, this is going to give you that strong foundation that will set you up for success, not just in your notebook, but also for the upcoming unit tests that eventually we will have. So let's get started. So what should we expect? Well, we're going to have our note check. We're going to do a phenomena study. We'll have a CER check, and some of this content will be on your unit test. So this video can be used as a review tool, but also your notes that are going to be based off of the PowerPoint, those will be the most helpful. And again, I'm going to encourage you guys to be very diligent and intentional with your notes. I'll show you examples as we go, and just keep in mind, you don't need to do it perfectly. You will get better and you'll find out what works the best for you as you go. But if you don't do it, it's going to be much harder. So take notes as we go. If you miss something or if you're worried about spelling as we go, don't worry. I will post the PowerPoint. This video will be posted and linked, so it's okay to have some gaps. You can also use your friends and other classmates to help fill in those gaps, or even check with me, and I will always help. But we don't need to pause class because we don't know how to spell something or to go back, because it will be available to you, okay? So let's start with the basics. What's a virus? Well, a virus is going to be considered a non-living particle, and it is made of genetic material. And that genetic material can be either DNA or RNA, which is a little different, but that's a unique component of what makes a virus a virus. And no, they don't contain both DNA and RNA necessarily, but they can contain one or the other. So their genetic material will be either DNA or RNA. They also have a protein coat, and that's going to be on the outermost part of themselves, and you'll see some examples. Viruses cannot reproduce on their own. That's why they need a host cell. They are pretty gosh darn lazy in terms of organisms because they're not an organism. They're non-living. They need that host in order to perform everything from reproduction to any part of their life cycle, because their life cycle is tied to the host performing all of those tasks. They are much smaller than cells. And that makes a lot of sense because a virus infects a cell. So it must be smaller. If it were larger, that wouldn't make a lot of sense. So that's just logic. They're going to come in a whole bunch of shapes. And a lot of times their names are going to correspond with their shape, like the coronavirus for COVID. It has what scientists like to call a crown-like shape. And so that sometimes you'll see that, sometimes you don't, and that's okay. We have different examples of viruses that we should be pretty familiar with just because we are a member of society. So we've probably heard the term HIV, influenza, coronavirus, and we love to abbreviate things because we're human. So coronavirus, we usually abbreviate to COVID, and there's different iterations or different versions, and that's where the numbers come into play for some viruses. The virus structure, well, it's going to have a capsid, and that is something that all viruses are going to have, and that capsid contains their genetic material. It's a protein shell, and it acts as a protective layer. The genetic material, like I already said, it's going to be DNA or RNA. And some viruses can have an outer lipid or fat layer, and that's going to be an envelope. So you'll see diagrams, you'll work with diagrams. This will make a lot more sense. There are surface proteins, and those are going to be around the virus, and those surface proteins help it attach to a cell when it's trying to infect the cell. So here you have just some hand-drawn examples of two pretty common types of viral structures. We have our spherical, and that's going to be the shape of a ball. It's a sphere. So inside is the genetic material. That genetic material is protected by the capsid, and all around this particular spherical example, you see little protrusions, little things that are sticking out of it, and those are going to have they're made of protein, and they make them a little sticky so that they can stick and attach to a cell. The envelope is going to be what wraps around all of this, and not all viruses have that envelope. Here you see a complex. In science, we love to name things exactly what they are. So a complex viral structure is going to look complex. This particular one has a little base plate here. It has some tail fibers. Those are the little spider leg looking things. It has a helical sheath. Those are pretty common. And helical is actually another virus structure. It's just not as common and it's harder to draw. So we'll look at some pictures of that. The neck would be up here, and right up at the top, you're going to see a capsid. It's labeled, and inside is genetic material. And if you notice, I specifically wrote genetic material instead of DNA or RNA because it can be a little generic. It's genetic material because viruses can have either DNA or RNA. So how viruses like to infect those host cells, I mentioned the proteins can make them sticky on the outside so that they can attach. So viruses must attach to a host cell. They can inject or fuse their genetic material into the cell. That capsid layer dissolves. It tends to degrade quickly, especially once that attachment has been made. Host cells are going to use viral genetic instructions to make virus parts. So once that virus enters the cell, it can start using the cell's organelles to start making copies of itself. And so that's where the reproduction and metabolism is going to happen. The virus hijacks the host cell. New viruses are going to assemble, and that means that they're formed, and then they exit the cell. This usually destroys the cell itself. So that can be part of the symptoms that you feel if you get sick with an infection from a virus. Having a lot of cell death is going to cause some discomfort within your own body. So that can lead to some symptoms. So we'll talk more about that later in the unit. Are viruses alive? No, they do not meet our criteria for what science would consider being a living organism. They don't have cells, they don't grow, and they don't make their own energy. They can only reproduce by infecting a host and using the host cell's ability to reproduce. And so this is why most scientists are going to consider viruses as non-living. So far, this is a great point. We've gone through things very quickly. What do your notes look like? So we're going to see my notes. Your notes do not need to look the same, but we're going to look at what I would do hearing and seeing this information. And if you want to adjust your own note page or make additions, now's a great time to do it. So we have sort of a title. We have viruses, the structure, because that's what I was focusing in on within my own notes. We have the TEKS, that's more what I consider important because it helps me make uh, a guide for what I want to make sure that we cover. The TEKS are what the state of Texas wants us to know about this. And I have some basic information. Viruses are unique. They must have a host. A host is going to be an organism that another organism lives within or on. And host is a very generic science term. So that's why the definition of a host is not specific to just viruses. You can have hosts within other parts of science, other parts of life science. So a tree can be a host for a bird because that's a bird lives in a tree. That would work as a host species. Um, so viruses, they need that host. A virus does not mean that it's living just because the definition for a host is an organism that another organism lives within or on. So the term host still applies though. Viruses are not cells. They don't have structure or membranes. They're lazy. I consider viruses to be a very lazy thing because they can't do anything themselves. They need to have that host in order to do anything. Otherwise, they're inert. They're not doing things. I have my hand-drawn versions of our complex and spherical virus, and I've outlined that there's genetic material, there's a capsid, and on the spherical, I've pointed out where protein parts would be for that attachment. So why do we want to focus in on not just the function of a virus, but on the structure? Well, their structure is one way that we identify them in science. We can look at them, and by looking at them, we can determine maybe how they can do their infections. Are they going to attach easily to cells and be a very high viral load in a person? Sometimes their structure, other than giving them their name and identifying features, it can give us other information. So it's one of those things that we use that structure to help us understand how they're carrying out their functions and also help us just categorize and classify them. So it's not something that is unimportant. I have some definitions for capsid and the differences between a capsid and an envelope. So the capsid is a protein shell around the virus's genetic material, and the envelope is the outer lipid layer that surrounds the capsid in some viruses because it's not present all the time. So the capsid, protein, the envelope, lipid. And remember, when we hit our biomolecules, these terms are going to come up again and again. So that's why I've underlined lipid and protein here. Genetic material can be DNA or RNA, and the main job of this lazy little virus is it wants to bring its DNA or RNA into a host cell, and then that host cell is going to transcribe the viral genome, and that's where the copying takes place, or how it takes place. So hopefully this makes sense. If your notes don't look just like this, that's fine. But the content should match up a little bit. Hopefully there's a lot of overlap. All right, so now let's talk about some real world virus examples. We have HIV, and this attacks the immune system. It targets your helper T cells. We have influenza that causes your seasonal flus. A lot of people tend to get vaccinated against it, and in your vaccinations for influenza, They tend to have 2 or 3 types or versions of influenza, and in science or your doctors, you might hear of like flu A or flu B. So there's different types of flu that can be tested for and also vaccinated against. So before the influenza season hits, scientists and doctors will build that vaccine, and they'll use what they predict will be the influenza that hits the most that year. COVID-19, we all have heard about this, and that's caused by a coronavirus. HPV is linked to some cancers. Rabies, this is a virus that affects your nervous system. It's more common in animals like bats, but it can infect a human as well. And just like a prion disease that also impacts your nervous system, rabies is a very scary virus, in my opinion. All right, so let's talk about some cells versus viruses, just so that we are very clear that there is a difference between what makes a cell and what makes a virus. So cells, these are living. They carry out life functions. They're reproducing, they have a metabolism. Viruses are non-living. They can only function with a host. Cells are complex. They will contain organelles, and this is true of both prokaryotic and eukaryotic cells. They're complicated. They have organelles inside them that are carrying out life functions. So there's going to be a cell membrane, mitochondria, cytoplasm, ribosomes, whereas viruses, they have genetic material, and that's going to be inside a capsid, and sometimes that capsid will be protected by an envelope, but it's not nearly as complex as our cell. Cells are in this uh, are going to be larger. We measure them in micrometers, and whereas for viruses, these are nanometers. They are much, much smaller. And remember, that makes a lot of sense logically because a virus infects a cell. And so it's going to have to be smaller than a cell. Cells can reproduce asexually or sexually. So you have binary fission, mitosis, or meiosis. Whereas viruses can only reproduce inside a host cell, and that's because they're hijacking or using the cell's own organelles, its machinery. Cells have a metabolism, and when I say metabolism, it means that it is producing and using energy. So that's what that term means regarding cells. Whereas our viruses, they don't have a metabolism. They can't make their own energy, which means they can't use energy unless they are using a host cell. So these are just some of the basics, the differences. And that's going to help us really make a lot of sense of our test questions that may come up. Remember, those test questions love to ask us small details. These are the details they're going to draw from. So a lot of times our test questions can sound more complicated, but if you look at your answer options, that might help guide you as well. For example, a virus cannot produce its own proteins because it doesn't have ribosomes. And we know it doesn't have its own ribosomes because who has ribosomes? Cells. Viruses have genetic material, a capsid, an envelope. It's not producing. And remember, ribosomes are one of those organelles that are very important in uh, metabolism and even cell transcription. So that's why if you look at your answer options, viruses don't have that because they don't have those organelles. They don't have any organelles really. So that's one way that use what you do know, even if it's at the very basic level, to help make sense of those test questions that might come up either on the STAR test or at the end of the unit for our unit test. So let's dive a little deeper. And remember, I mentioned ribosomes, and there's a reason I'm talking about ribosomes, because ribosomes are needed for cell growth and metabolism. Can a virus grow or metabolize? No, not without a host. So ribosomes are crucial for our protein synthesis, which is why whether you're a prokaryotic cell or a eukaryotic cell, you have ribosomes. So both viruses and cells can have nucleic acid chains because that's just genetic material. But a virus uses its host cell to replicate because a virus lacks ribosomes. So this is just one way that we can take a moment, make sure we are not just mindlessly taking notes because we need to be intentional about this. You know so much about all of this. Make sure your brain is processing as you go. So don't stress out, it's just notes, and there's always reviews. But I want to make sure that you're being intentional. So that's why I'm drawing your attention to certain things. Not just because it'll show up on a test, but because all of this builds. So the more you know, foundationally speaking, the easier it gets as you climb that pyramid up. What do your notes look like? And it's okay if your notes don't look like mine. So we're going to take a minute, look at some of my notes. I know I have terrible handwriting, but notes are for me. I'm going to review them. And so it's okay if your notes are not picture perfect. They do not need to go on Instagram. So there's meant to be a tool to help you. So let's take a minute and just think. Remember, your notes don't need to look like mine, but what do you notice about my notes? Well, I have another title, Virus versus Cell, because that's kind of the focus of this page of notes. I mention my TEKS again, compare the structures of viruses to cells, because that's what I personally like to help guide my notes. And then I have cells on one side, I have virus on the other, and then I have some of those tasks that who does what. So for reproduction, a virus needs a host, whereas cells do it by cell division. Cellular structure, viruses do not have, but cells do. They have organelles. Genetic material, well, cells are going to have that DNA, usually, and our viruses, they can have DNA or RNA. Their size, viruses are teeny tiny, and cells are going to be larger. Are they living? Cells are not living. And viruses are non-living. Cells are living. I misspoke, but hopefully you guys followed the pointer instead of listening to my cuckoo bananas. Some of those characteristics, well, we have the presence of a capsid, and we have the presence of nucleoids. So cells and viruses are different structurally and functionally. And I have on my notes just some background info that hopefully y'all remember from 6th grade, and maybe 7th grade they hit it again, but usually this is from 6th grade. And that's just that cell theory. What makes, or what do scientists use to classify these living things and cells as a living thing? So in the 1830s, we had Schwann and Schleiden, I never know how to pronounce that. And their theory of cells gave way to that modern cell theory. And modern cell theory, all known living things are made up of cells. The cell is the structural and functional unit of all living things. All cells come from pre-existing cells by division. And the flow of energy occurs within cells. So that metabolism part that we talked a little bit about. And so that background you wouldn't have necessarily because I didn't talk about it. But that's something that I hope you remember from 6th grade when it would have been covered. And if you're like, I know that, that's fine. Great. If you're like, that's okay. Just review. You can write this down now. You can even make a note, like go back to cell theory, review that. So why did I bring up cell theory? Because cell theory doesn't actually explain how viruses require the use of cells. Viruses are weird. That's part of their uniqueness. They're weird. And so cells are living. Viruses require cells in order to survive. So it's one of those like wild things. I love it. Science is filled with things that hopefully you guys will come up with answers for when you enter the scientific field as professionals. So now we've reviewed a whole bunch of basics about viruses. So now we're going to think about our phenomena study. And you would have done this last year. And so we're just going to ease back into getting used to doing our phenomena studies. And I'm going to use HIV, epidemics, and pandemics, and the differences between epidemics and pandemics to kind of talk about a virus like HIV. So this is just going to be part of a check I do. So yes, you have to pay attention. And yes, I need your thoughts on this. So our phenomenon prompt. In the early 1980s, scientists noticed clusters or groups of people with weakened immune systems. They weren't connected by location or behavior, but their immune systems were failing. Today, we know the cause. It was the HIV virus. And there's some questions to help get you thinking here. Why did it spread differently? Because remember, it's in clusters. Why was it hard to detect? Well, these individuals were not connected. It was happening to just pockets of people all over, and they didn't share the same exact symptoms other than a failing immune system. So these are a lot of ways that doctors really made groundbreaking research in a short time period if we think about it. In the 1990s, there was globally a lot of people unfortunately dying from HIV, but it's started to be noticed in the 1980s. So within this short time period, doctors and scientists noticed a trend and they had to work together to kind of figure out what was causing this. So that's your prompt, a little bit of background. And remember, with a phenomena prompt, we want to have things that we notice about it and things that we wonder about it. So here's just a little bit of a recap because I don't expect you guys to be an expert, but HIV infects the cells that coordinate the human immune response. Remember I mentioned HIV attacks your immune system? So when these cells are killed or compromised, and by compromised, they're in the process of dying, your body cannot effectively protect itself from any infection, which means individuals with an active HIV infection, they're going to start getting sick from a lot of things, from fungal infections to the flu to pneumonia, all sorts of what might be serious or not serious infections are going to start attacking the body because there's no natural immune response. And HIV does this by infecting the helper T cells. So HIV makes the body vulnerable to all those secondary infections. So you're not just sick with the HIV virus, you're going to then get sick because your helper T cells are sick and dying. You're going to get sick from all these other infections. So that's just a little bit of background, a little added information. So now we have, in your own words, I want you to write in your notebook an I notice and an I wonder. Those notices are things that you are noticing. So from the prompt, from the recap, what did you notice? What was presented? Notice statements are going to be very straightforward. You don't need to do a lot of heavy lifting in terms of your brain, whereas your I wonder statements are meant to be questions. They do not need to have an answer, or they might have an answer, but that answer requires some further investigation. So I notice these are going to be very direct, whereas your I wonders are your internal questions, things that you want more information about. They can also be questions that don't have answers, and they can be questions that we just have to do a little bit of work to get the answer. So take a minute, make sure you write this down in your notebook. Okay, if you're still writing, that's okay. Let's do a note check. What do your notes look like right now? And you should have some notes, quite a bit, in fact. Do your notes need to look identical to mine? Absolutely not. In fact, I hope they look different. So on this page, I have my I notice. HIV infections sound generic. There doesn't seem to be an obvious early symptom for doctors. Okay, so that's what I noticed from the prompt. What do you wonder? How did doctors connect the dots? That's something that I think is very interesting to think about. I asked a question based off of the information presented in the prompt, whereas my I notice, I use the prompt information to make a statement about the information provided. So do you see how there's a difference between these? And do you see how intentional you can be with this? And it's not just something to ignore or think is silly. So these phenomena studies are there to help make a connection between you and the real world sometimes. It's also meant to make a connection or bridge the information you're being presented from the textbook, from my presentations, to yourself. What are you thinking? How are you interpreting the information? So this is a bridge between what's presented and what you're figuring out, what you're noticing. So that's why these are important to do and to take seriously. And there will be an I notice and an I wonder check at the end. So this isn't something to ignore to do. Please get it done. I notice, I wonder. And this is so important because we're going to revisit this. I want you to make sure that you have a very firm idea of what my expectations are going to be. This is early days. We are still getting used to being biology students in the biology classroom. So there are ways that I'm going to expect you to continue to elevate your own thinking within this, and I will help guide you to that. So these are just some deeper dives for your I notice and I wonder. That's based off of a HIV phenomena prompt. So some diseases spread quickly. HIV spreads differently. It's not spread through coughing or something like that we might see with influenza or coronavirus. Symptoms may take longer to appear, so not all viruses are going to display symptoms in the same way or same time frame. Some of these wonders, why doesn't HIV show those symptoms immediately? What is a difference between an epidemic and a pandemic? So we're going to talk about that. This is that nice shift into getting more information, using more science vocabulary. And also just it's okay to have big questions that are hard to answer or don't have answers. Why was HIV so hard to understand? So you can see there's a variety of ways to interact with these phenomena prompts using the same prompt. And so I also want you to notice every single wonder is a question and every single notice is a statement. So we want to make sure we do not confuse these. So let's use HIV as a viral review because I told you these phenomena studies are meant to bridge a gap, to make more connections. So HIV is a retrovirus. And I didn't mention this before in the phenomena study, but I could have. HIV is a retrovirus. And remember, viruses can have either DNA or RNA as their genetic material. Only, and I use only very sparingly because it has to be an only statement. Only and all stand out. Only in a retrovirus where the genetic material is RNA will the molecular process of reverse transcription take place. So most cells are going to copy DNA, RNA, protein. That's just how it goes. In retroviruses, it goes RNA, DNA, RNA, protein. And this is the big difference, and that is what makes the retrovirus HIV, it's using reverse transcription. This is unique. Not all viruses are retroviruses. HIV just happens to be one. And so a retrovirus is going to do reverse transcription, and that reverse transcription does not start with DNA. It starts with RNA. After RNA starts things, though, it follows the same process. So, reverse transcription, RNA, DNA, RNA, protein. DNA, RNA, protein, that's what cells are usually doing. So this is just one way we are incorporating new vocabulary, and we're using HIV as a tool to incorporate some of that new vocabulary like reverse transcription. It's also a way to talk about how viruses have genetic material that can be either RNA or DNA. But at the end, they still need that host cell to make those copies, and that's their end goal. They want to have copies of themselves. So please make sure that you are acknowledging that reverse transcription is taking place within the HIV retrovirus, and it starts with RNA. And it's easy if you think about retrovirus, R, reverse R, and RNA. So you got your R's. So why do I need to know this? Well, sometimes history likes to repeat itself. And it doesn't mean that history is always repeating itself, but it does tend to do this. So, in 1918, an outbreak of influenza or flu infected over 500,000 people all over the world. Many of those infected died, unfortunately. In 1994, an outbreak of pneumonic plague spread quickly through the Indian city of Surat. Over 50 people died. So the 1918 influenza outbreak, this was a pandemic, whereas the 1994 plague was an epidemic. These things are going to constantly come up and you'll hear them in the news, and you want to make sure that you're using this vocabulary correctly. So we have our why do I need to know this? You are going to have to interact with society as a human being, and you want to know what the words mean when they're being used. So a pandemic, this is worldwide, and it's a lot of people. An epidemic, this is not going to be worldwide. So think about your HIV case study, that phenomena study that we did. Was HIV a pandemic or an epidemic? Was it worldwide? Yes. So because it was worldwide, that means HIV was a pandemic. It was not localized to one geographic area like an epidemic. An epidemic is going to be something that stays in one area for the most part, whereas a pandemic, it's going to impact either the whole world or a large part. A pandemic taking over North America, that would count because it's such a large portion of the globe. An epidemic is going to be in like a city, like that pneumonic plague outbreak in 1994 that was mentioned. So, why do I need to know that history sometimes repeats itself? Because we want to make sure that we are being precise with our language. As these scientists, we don't want to be lazy. And this is one of the easiest ways to not be lazy. A pandemic is worldwide. One way that you can make sure that you remember this, draw a little world next to it. Write world. And it is not the same as an epidemic. An epidemic is going to be geographically like a condensed space, one spot. And sometimes you'll hear very similar sounding words and similarly spelled words in science. And that's going to be like epidemic and endemic. Well, an epidemic is something impacting one geographic area and it's contained there. It usually has a start and an end point. This is like the pneumonic plague outbreak in 1994. Eventually it ended. It's not still ongoing. Whereas endemic is going to be more similar to how malaria is constantly present in certain parts of Africa, Central Africa. So endemic is something that impacts one area all the time. It doesn't go away. So that's like malaria. Whereas epidemic, it's going to be something that's impacting one geographic area and it has a start and an end. So some of our key vocabulary, we've mentioned HIV, we've mentioned virus, host cell, immune system, epidemic, pandemic. These are just vocabulary terms that we've used. I want to make sure you all are very, very firm on at least all of these, but make sure you know the difference between the last two. So our epidemic and our pandemic. Know the difference. So pandemic, it's everywhere, worldwide or potentially worldwide. Epidemic, it's in a specific area. All right, here is your CER writing prompt. I want you to do this in your notebook. Please answer the question being asked. Please use the evidence that makes sense. And I want you to really focus in on your reasoning here. Okay? So I look forward to reading all of these. Spend about 10 minutes, no more, maybe 15 minutes max. All right. Have a great day.